Good evening, warm welcome to all of you. My name is Patrizia Gambarotta. I am the director of the Italian Cultural Institute, and I'm talking to you from one of our classrooms where we usually have our Italian language courses. Right now our courses are online, so unfortunately there are no students with us. Professor Cavalieri is joining us from Florence in a minute. I would like to thank the Federation of Finnish Learned Societies for organizing the Night of Science and for asking us to join them for the third year in a row. Duccio Cavalieri is a microbiologist and geneticist. He firmly believes that we can understand the complex functioning of biological systems only under the lens of evolution. His main research interest is the application of genomics and metagenomic approaches to the study of the role of microorganisms in human evolution and in the functioning and evolution of complex biological systems. The title of his lecture, the full title, is Alcohol, Yeasts, and Human Evolution. Everything you wanted to know about yeast but were afraid to ask. And I hope by the end of the lecture you will not be afraid to asking questions because we have about 15 minutes for Q&A. So let's welcome Professor Cavalieri.
Hello everyone, buonasera a tutti da Firenze, Italia. Hello everybody, can I start?
Yes.
Hello?
Yes.
Good. So I wish we could be present in person after this long time of isolation due to COVID. And today's theme will hopefully reconcile you with microorganisms, at least with one part of the microbes that inhabit us. So the title is a challenge. The title is also, in a way, one side of the truth about the role of alcohol and fermented beverages in facilitating, shaping, and anyhow influencing human evolution. First of all, I want to say that a lot of the things I will try to discuss with you this evening are becoming clear and evident, are becoming known because in the past 15 years we have been learning really a lot about something that we could not see under our eyes except if we could grow them in a petri dish. And this is the microbial world, is the incredible diversity of the microbial world. Here on the right, you see a deformation of a petri dish where you see the different shapes that bacteria get. But we have to remember that microbes are usually have a size that is less than 10 micron, so we can't see the microbial cells with an eye. We need microscopes. In the past 15 years, instead of using lenses in our microscopes, we are using sequencing. And we are using the generation of sequencing machines that have been used for sequencing the human genome. Your island is famous for having participated in the largest genetic screening that has ever been seen on a population scale. And I would say that so the sequencing technologies applied to the human genome are now being applied to microbial communities, those microbial communities that inhabit our body. And it is generating an enormous amount of data. This is also changing the vision that we have of ourselves as humans, because we have in our body on average we have 10 times 10 to the 12th human cells, and we are inhabited, colonized by 100 times 10 to the 12th bacterial and fungal cells. So for every human cell, there are 10 microbial cells. If we move it at the genetic scale, we see that while the human genome is made up of 30,000 genes, the human microbiome, so the genes of these microbial communities, account for 1 million genes. So we could say that bacteria, fungi, and viruses make much more of our self than our own self. So we are really a consortium of different sequences. This is even more true if we consider the amount of RNA and DNA present in 1 ml of human blood. If you sequence the amount of DNA present in human blood, you will find that 3.6% of the sequences, less than 5%, are human sequences. 4.4 sequences are not human, but are of different, like fungi or protozoa. 9.5 are bacteria. 67.7% are viruses, and then around 15% are unknown. Why did I put up this slide? I did put up this slide to tell you two things: that for our existence and during the entire time of our evolution, we have been dealing with the interactions not only between human cell and human cell, but between human cell and bacteria, human cell and fungi, human cell and viruses. Therefore, you should not be scared of an RNA vaccine like the ones that are being proposed right now, because we see viral RNA since the birth of our existence on Earth. Okay, so this is part of ourself in any way. So if you pose yourself the fundamental teleological question, who are we? What is human? One of the answers that we have now, we are nothing but walking hologenomes. What is an hologenome? An hologenome is the entire structure of the genes, proteins, and networks of functions of our cell, bacterial cell, fungal cells, and viral cells. And from a Darwinian point of view, the unit of Darwinian selection is the host and its microbiome. Now, when people ask me who I am as a scientist and what is my field of research, I like to say that I am interested in the study of anthropomicrobiology. What means anthropomicrobiology? It means I'm interested in trying to understand the connection, the interconnections between the microbiome or microbial world, ourself, in an evolutionary space. And this requires to see which are the mechanisms that we have developed to deal with these microbes and interact with these microbes, exploit them, but not being killed, grow and flourish with them without being harmed. And these mechanisms indeed have an intermediate, that is diet. What we eat is the strongest force that shapes our human microbiome. We can change our microbiome by changing diet, and the fact that we have changed diet has been corresponding to the fact that we have uptaken different classes of microbes from the environment, because the major source of the microbiome is our mother at the birth. Yes, women count more than men for inheriting the microbes to the child. And then during weaning and during growing, the environment and the food. One of the greatest anthropologists that we have known said, "We are the heirs of inherited characteristics accrued over 2.5 millions of years. The vast majority of our biochemistry and physiology are tuned to life conditions that existed much before the advent of agriculture, some 10,000 years ago, the advent of agriculture. Thus, genetically, our bodies are virtually the same as they were at the end of the Paleolithic era, some 20,000 years ago. What this means is that we are adapting to a rapidly changing environment much more through the genes of the microbes and their products rather than by changes in our own genome. In particular, the modern cosmopolitan transformation that all of us we are facing in the Anthropocene has impacted our ancestral mutualistic relationship, the ones that we have established 10,000 years ago. It's like saying that 10,000 years ago we have been striking a deal with classes of microbes, and during this recent evolution, this deal has been broken. Breaking this deal has led to many disorders, and therefore, trying to reestablish this equilibrium is one of the major tasks of the anthropomicrobiology. Yes, everybody of us at the moment is witnessing the rise of anthropomicrobiology as a science. If we look at the evolution of human diet, we see that during the Paleolithic era, from 2,300 to 2,300,000 years ago, we have been seeing the passage from Homo habilis, Homo erectus, Homo sapiens. We used to be, in the beginning, scavengers. Then we were hunter-gatherers. And only recently, during the Neolithic era, around 10,000 years ago, we changed drastically our lifestyles and we started husbandry and sheep farming. We started agriculture. We started accumulating products of the agricultural production, and we went into the metal age. This passage didn't happen only thanks to us. This passage was witnessed and followed by the genes that our microbes have been carrying. And one of the major questions that I posed when I started presenting this lecture, why ethanol? Because the means of selecting the microbial communities that inhabit our bodies, the molecules that help this selection, are as important as our immune system to shape our evolution, seen as the interaction between our genes and the genes of our microbes. Thus, during this lecture, we will hear the words of Saccharomyces, Saccharomyces cerevisiae, Clechera, Schizosaccharomyces, Pichia, and Senula, these are yeast, and bacteria like Lactobacilli, Bifidobacteria, and Prevotellacea that are shaping our diets and our evolution. We started this trip, this adventure, many years ago, at least scientifically. And I remember 10 years ago, when we were thinking about the right experimental setup to starting to study what was our ancestral microbiomes and how we evolved with them, we tried to see whether we could find some evolutionary relics that were going back to 10,000 years ago, to the passage between the hunter-gatherers lifestyle and the lifestyles of the farmers. And what we witnessed and decided to study was the Bulpon village in Burkina Faso in Africa. This is a village that lives on an economy of subsistency. So the population size is determined by what the ground produces. The instruments used to produce the ground are quite primitive in a way. Therefore, what you raise is sorghum, millet. It's very hard to raise cereals and grains, but some of them do grow. And the lifestyle is a community lifestyle where people prepares food all together. This is the communal kitchen of the village. And the food is prepared. This is a termite's nest. It's prepared indeed not in a sterile way. So one of the questions was how you deal with this. It is a healthy situation. What we observed immediately, we were looking at children, is that the microbiome, so the microbial communities of African children living in these, I would say, very low hygiene conditions, the microbial communities of these kids were incredibly healthier than the microbial communities of our kids, and were enriched of microbial species that were producing molecules that are important for maintaining the gut health, like short chain fatty acids, acetic acid, propionic acid, butyric acid, valeric acid. Now, think about this. This is where we passed. I mean, if you look at the village of Bulpon, you find things like this for grinding stones. You find things like this for working the ground. So we were really in a parallel that would fit. And therefore, what this means is that the Neolithic Revolution 10,000 years ago has been dramatically changing the human lifestyle. We have seen the advent of cereal cultivation, we have seen plant domestication, we have seen animal husbandry and farming, and all of this, for the first time in human history, has led to the accumulation of food products, seen as accumulation of grapes or apples, of grains, sorghum, barley. This accumulation of food products had to be maintained because these food products in excess would rotten, and the rottening of the food products could lead to disease because it could lead to gut infections. So one of the major questions was how to preserve the fruits of the harvest, how to preserve and how to prevent the rise of infectious diseases. Well, an evidence of the strategies used for this preservation comes from reading some of the old Roman history and Greek tradition history books. One of them is Theophrasto. Theophrasto, whose book Enquiry into Plants We Have Inherited, mentions how odors of decaying animals and plants are evil and putrid, except for the aromas of wine, because fermentation is necessarily to achieve wine's health benefits. So Theophrastus at the time offered many observations on wine growing techniques, including matching salt types with different varieties, vine training, and even reducing yields to make high quality grapes. But the whole deal was how to preserve the nutritional potential of grapes, as an example, through wine, through alcohol. Now, our history goes down and sees what's the evolution of plant domestication. If we look at the areas where plants like apple, where the vine, the sorghum, millet, but also where cereals have been domesticated, we see that this goes down to rice, it goes down to early urban China, early urban Mesopotamia, and the area that currently corresponds to Israel, Syria, and Turkey. What is interesting is that the areas of urbanization, so gathering together in villages and cities, correspond to the areas of plant domestication. What this means is that people is realizing that investing on increasing the production of fruits and of vegetables results in an increase in human health. And when we look at Vitis vinifera, we see how the development of Vitis vinifera cultivation has slowly been changed and affected by the preferences of the human societies, in particular the Roman societies, related to wine taste and alcohol content. Marcus Aurelius Probus, a Roman emperor that lived 2066 to 2022, imported some specific vines from the area that currently corresponds to Romania, it's called Thracia, to the area around Cuma and near Naples, and crossed them with Falerno, with the existing Italian grapes that had been raised till then by the Romans. This cross basically gave rise to Cabernet Franc, that when was crossed to a grape type present in Gallia, gave rise to Cabernet Sauvignon. So we have been selecting these the plants. We have been selecting the plants for improved the taste and improved wine production, but this has led to another selection. This has led to the selection of something invisible. This has led to the selection and domestication of a microorganism, Saccharomyces cerevisiae. Let's look at the history of the fermentations. We know that by 6000 before Christ, the Babylonians, remember Babylonia is one of the two sites with China that sees the first domestication of plants. Babylonians were drinking beer. We're drinking beer and they're doing beers basically by batch culture. They were using a previous batch to inoculate the second batch. And this goes on in Sumeria, Egypt, India, and China. And by 4000 before Christ, the Egyptians were baking leavened bread, and they were not doing it by using beer, but they actually were using a Saccharomyces cerevisiae that was coming from grapes. Who is Saccharomyces cerevisiae? Saccharomyces cerevisiae is a eukaryotic mechanism. This is the beautiful shape of Saccharomyces cerevisiae. These are Saccharomyces cerevisiae cells. It has been used in genetics for many years because it gives rise to unicellular colonies. It's actually has a sexual cycle. Saccharomyces cerevisiae has invented sex. It's the microorganisms. One of the first microorganisms in the fungi kingdom that divided the sexes into A and alpha, like males and females. And so it has a haploid life cycle, the gametes, and there is a diploid life cycle, in which basically it's what you see, the vegetative life cycle that is classically considered for higher eukaryotes. Colonies of yeast can be like normal, like being smooth, or can have incredible shapes like the one up here. And yeast can live as single cells or start making clogs and producing multicellular structure. As I've said, yeast products in human history go long way back. I started studying the evolution of yeast when I was in Harvard. My lab's motto was my first appointment as an independent researcher was in vino veritas. I used the Harvard logo. It was kind of forbidden at the time, but it was something between friends. We know that in the Near East, by the time of the book of Genesis, perhaps at least 10,000 years ago, we see the first written descriptions of winemaking. And we find evidence of the winemaking procedures, like the harvest, the carrying of the grapes using animals, and then the crushing of the grapes and the fermentation, from some beautiful paintings in the Casa dei Vettili in Pompeii in Italy. And we have some great witnesses from great poets. I mean, in Vino Veritas comes from, used by the Lacing, comes from an Egyptian motto that says, in water you see your own face, but in wine the heart of its garden. It's an ancient Egyptian proverb. And Homer, that wrote the Iliad and the Odyssey, said no poem was ever written by a drinker of water. I mean, this is not an exhortation to drinking, by no way. I mean, I will clearly state that alcohol is not good for you. We should drink intelligently, but we cannot deny that the evolution of wine fermentation has led to the evolution of wine society, really from the times of the Sumerian Empire. This is the content, the chemical analysis of a bronze situla. Is like a cup of bronze used to eat, found buried in the King Midas tomb in Gordion in central Turkey. There was a barbecued lamb and goat seasoned with Mediterranean spices, assorted legumes, and kicheir. It's mixed fermented beverage made from grape, wine, barley beer, and honey mead. What this means is that at that time, everything that could ferment was fermented together and used as a way to preserve the energy that was present in these nutrients. Now, my lab was lucky enough to be exposed to these to the discover to I think one of the fundamental discovery in the study of the evolution of Saccharomyces cerevisiae. Uh, when I was at Harvard, we received from Patrick McGovern, an archaeologist, we received samples that were coming from the tomb of King Midas. King Midas was a pretty nasty king, so one of the last kings before the dynasty of the pharaohs, and he was buried with hundreds of wine jars that were coming from Galilee. This is King Midas' tomb. There was a specific chamber that was the wine cellar. Uh, the Scorpion King is well renowned for have been the inspiration to some trash movies of the end of the 90s, but has been also is very well known for having unified the Egyptian tribes of the High Nile. And what we saw, we were able to extract DNA. These are the extraction from some of these jars, wine fermentation of these jars. And using a technique called polymerase chain reaction, we were able to amplify yeast DNA and sequence it. And we characterized Saccharomyces scorpionis, was an ancient yeast in a way. So was the last remaining DNA of one of the most ancient fermentations that we could date. Very recently, last year, there was another paper that studied the feces of Hallstatt miners from a people that goes from the Bronze Age, I would say around 1,500 years before Christ, to modern time. And this has been possible because the Hallstatt mine is a salt mine where basically feces remain stabilized in time. These paleofeces were extracted, and the scientists, doing an interdisciplinary approach based on DNA sequences, proteomics, and metabolomics, characterized sex and haplogroups of the Hallstatt mines, the gut microbiome, presence of parasites, the animal diet, the plant diet, the fermented foods, and the fermented beverages. And what the scientists discovered, they discovered that the tradition of our ancient microbes is very similar to, let's say, what we were discovering in Africa. So the Hallstatt miners had mainly a vegetarian diet. They were drinking a lot of beer. They were being exposed to Saccharomyces cerevisiae, Cluveromyces marxianus, because also with beer, they were drinking probably the equivalent of kefir, but you could also find some of the microorganisms that are currently found in herbaceous cheeses like probably gorgonzola. Since we are in Italy, I would rather think of gorgonzola than other cheese consumption. So we could resurrect an ancient diet using DNA-based analysis. Why these diets were so important for human evolution? These diets were so important for human evolution because if you have to tell which was the principal cause of death in Roman times or during medieval times or at the age of the Hallstatt miners, the principal cause of death was gastroenteritis and gastrointestinal infections. And you know how, let's say, the Gauls had defended Alesia. What we know about Alesia, and when Caesar puts the siege to Alesia, we know that Caesar constructed a series of fortresses that surround the area where the Gauls are located, where the stronghold of the Gauls are located. And why this stronghold was impregnable was not only because the Gauls warriors were so fierce, but it was also due to the fact that the walls warriors used to poison the waters in the area. It said that Alicia was surrounded by marshes, and by killing animals and putting them into the marshes, if we would drink the water from the marshes or from some of the rivers or some of the springs in the area, you would get gastrointestinal infection. And so the army of the assailants, the invaders, would be killed not by the spear or by the arrows, but would be killed by gastrointestinal infections. Maybe this is why the Romans, when they moved in their campaigns, used not to bring not water tanks, but they would bring wine tanks. They would bring grapes, they would bring olive trees, and as soon as they had established, they started conducting aqueducts. Drinking safe water was something essential for Romans, as well as was for many of the Nordic populations that were living in Belgium or in the Netherlands or in Denmark, that would rather drink beer than water that was unsafe. Therefore, one of the first applications of human evolution of wine is that wine maybe was one of the elements that allowed the Romans or Alexander the Great to conquer such large parts of the world. Actually, Alexander the Great probably died of wine intoxication and wine pollution, so it also exposes you to the risk. But you have to know that Roman soldiers would never drink wine alone. They would dilute wine 1 to 3 with water. Wine alone was allowed only during the bacchanals, that was like these religious holidays that Rome had seen, or were drink pure only during these banquets where people would get drunk, but otherwise the average Roman soldier would not go drunk in a battle, but would dilute 1 to 3 or 1 to 4 the wine to make water drinkable. Now, all of these that I've told you existed and raised and started when humans had no idea of what a microbe is. And that's because the microbes were very small. So the history of the study of Saccharomyces cerevisiae starts in 1680, thanks to Antoine van Leeuwenhoek, that is a Dutch scientist and merchant that constructs the first solar microscopes. Here there is a lens. Here you can put a piece of the things you want to observe. And then with this method, Antoine van Leeuwenhoek described white blood cells, red blood cells, yeast cells, sperm cells, some bacterial cells. The association and the discovery that Saccharomyces cerevisiae is the actor that makes wine has to wait to 1837, to a French scientist called Cagnard-Latour that discovered that yeasts are responsible for fermentation and described Saccharomyces cerevisiae as follows: little animals that suck sugar through their snout and emit ethanol from their intestine and CO2 from the urinary apparatus. So it's a total humanization of the fungus. And this is really the beginning of the revolution of microbiology, because in 1839, Justus von Liebig and Friedrich Wöhler demonstrate that alcohol, ethanol, is produced by yeast, demonstrate that yeast is the main actor that makes beer, both Lambic beer, Carlsberg beer, or the major beers, and in Denmark, Jacobsen, that is the founder of the Carlsberg brewery, brings Emil Christian Hansen, one of the most renowned scientists of the time, a biochemist, to the institute that is started under Carlsberg. And Christian Hansen develops the first pure yeast cultures, a strategy to maintain them, and started to study the biochemistry of fermentation. And in parallel with Buchner, discovers that alcohol can be also produced by yeast extract. The fantastic part of this story is that at this time, Louis Pasteur gives us the best definition of science. Louis Pasteur is really a genius. He's a scientist, and someone did applied science and fundamental science. And Pasteur says il n'existe pas une catégorie de sciences que l'on puisse donner le nom de sciences appliquées. Il y a la science et les applications de la science liées entre elles comme le fruit et l'arbre qui la porte. This means that it doesn't exist applied science and pure science. There is science, and good science is like makes good fruits. If you don't take care of your tree, if you don't fund science, well, you will never get fruits. And this is, in my opinion, one of the most fantastic definitions. So yeast became the main actor of the scientific revolution that led to several Nobel Prizes. The Nobel Prizes for Physiology and Medicine, Lee Hartwell, Tim Hunt, and Paul Nurse. Hartwell working on Cerevisiae, Nurse on Pombi, and Hunt on Spongy. Yoshinori Ozumi in 2016 for the discovery of autophagy, the mechanism through which the cells stay young, and then the telomeres from Elizabeth Blackburn. And it was so important that basically also jazz people making music, realized the importance of yeast in fermentation. Carmen McRae, that is a jazz player, said blues is to jazz what yeast is to bread, without it it's flat. The person here is Andrew Murray, that was the fourth person that should have had the Nobel Prize for the cell cycle, has been my mentor when I was at Harvard that actually gave me this slide. So again, science moved back from the bench to the field. And in the past years, in the past 10 years, we have been using Saccharomyces cerevisiae as a model to study population genetics, but also to understand what is that makes wine and fermented products so attractive for us. The study of the Saccharomyces cerevisiae genome from wine and beer showed the first evidence of domestication and demonstrated that while the evidences of domestication in wine yeast are uncertain, so wine yeasts are very wild, I would say. For beer, there is indeed a clear evidence of human intervention. So during beer production, without realizing what we were doing, we have been creating sterile hybrids of Saccharomyces cerevisiae and Saccharomyces paradoxus, and Saccharomyces cerevisiae and Saccharomyces baianus. So not only we have selected which strains were the best, but we have also led to stabilizing them by reaching sterility through interspecific hybridization. And all of this was done in Belgium, in monasteries, without people even having an idea of what yeast would be. And the other interesting story is that yeast have been following the fluxes and the movements of the men, commerce, selling goods that are produced by cultivation, transporting wine, transporting grapes, transporting barley, transporting fermented wine. The Phoenicians were some of the first to bring wine through the Mediterranean and exchange wine and grapes for iron, for swords. And during this passage from China to Europe, yeast has not only been used to make wine, but has also been used to make cheese, like kefir. Kefir is a combination of Saccharomyces cerevisiae, Saccharomyces cluveromyces lactis, and lactobacilli. So communities that see many different microorganisms. And as I said, this incredible evolution of science and of human history has been really driven by the fermentation process. The fermentation process that Saccharomyces cerevisiae ferments sees a strange strategy. In the beginning, when there is a lot of sugar, Saccharomyces cerevisiae decides to use a procedure that to ferment the glucose that produces 1/16 less energy than respiration. Decides to ferment. It's like saying, I throw my money in the bin because I really want to make something that is not as efficient as it could. And I do that because I'm trying to select the production of ethanol, a toxic molecule. Ethanol is toxic for bacteria, kills bacteria, is toxic also for humans. And when it goes over, let's say, 18-20%, is also toxic for cerevisiae. But cerevisiae is really a good drinker. It withstands up to 20% ethanol. And when ethanol is too much and the glucose is exhausted, then we switch. Instead of transcribing the ADH1 gene, we transcribe the ADH2 gene and we convert ethanol in acetaldehyde and then in secondary products of fermentation. When people was looking at yeast and their applications for wine production and beer production, saw that Saccharomyces baianus, Uvarum, Cerevisiae, Pastorianus, and Paradoxus are those that we use the most in wine fermentations because they resist, produce, and survive at concentrations of ethanol that are greater than 8%. A concentration of ethanol greater than 8% allows you to kill a lot of other fungi that could steal your sugars in the grapes and to kill bacteria. So when we look at history of yeast evolution through a humanized point of view, we realize that we could see wine in a different way. Wine is the end product of Saccharomyces cerevisiae victory in the struggle for the survival of the fittest. In fermenting natural sugars, Cerevisiae produces ethanol and acetic acid to suppress competitors. So the thing we like so much, ethanol, in the end is a toxin. And it's toxic also for our liver that has been developed, whose the production has been evolved to wage chemical warfare from Saccharomyces cerevisiae on all the other microbes that are present on the grapes and that are present on the crushed grapes. In fact, the production of these toxins as weapons to suppress competitors is self-limited by the yeast's own potential to resist and adapt to them. And the selective pressure on the ability to produce and adapt to stressors is the forerunner of strain diversity and evolution. This is the rate of production and resistance of ethanol in acetic acid from 433 yeast strains from Chianti, and you see that the average yeast strains, when you give them 18% glucose, they get to 14% ethanol, but there are some of them that go over 15%. And also here is acetic acid, is the counterbalance. We are between 600 and 800 milligrams per liter. The proof that this is a killer for other fungi and other bacteria come, let's say, from this work here. The line here tells you the raise of the fungi during a fermentation that lasts for months, because it's a nice vine from Trentino. And this is the bacterial fermentation. So here is the crush of the grapes. Bacteria are much more than yeast. This is quantitative. Bacteria go up, yeast go down, but at a certain point, slowly, after 2 weeks in the cold, some of the yeast, in particular Baianus, starts growing, start producing ethanol. The ethanol goes up, the fungi goes up, the bacteria go down, and in the end, the bacteria die after 80 days. Now, all of this starts from a paradox. We have been giving for granted that basically fruits carry Saccharomyces cerevisiae. But it's very interesting that a few years ago, when microbiologists went and looked for the presence of Saccharomyces cerevisiae on the pristine grapes, the intact berries, they wouldn't find none. And therefore, the idea was that cerevisiae is so associated with humans that it stays only in the wine cellars. So it's so associated to human evolution that the way we produce wine provides the best place where yeast stays during the winter. Yeah, that's true. I mean, wine cellars, beer making cellars, they do have Saccharomyces cerevisiae. But the Romans, in the beginning, didn't have wine cellars. The Egyptians didn't have wine cellars. So yeast had to have a natural environment, and So a few years ago, Mario Polsinelli and Bob Mortimer, two of my advisors here from Florence and from Berkeley, discovered that if you look at pristine grapes, basically the probability of finding Cerevisiae is 0.0%, but if you look at broken berries, it's 25%. And now the question is, who is that brings Cerevisiae on the berries? Cerevisiae is very heavy. It doesn't fly with the wind. It doesn't move with the wind. It doesn't stick in small particles of air. It's quite heavy, so it stays where it is. Needs a vector. So we started looking for the vector, and by a simple of serendipitous observation of an animal that was eating a berry, we started hypothesizing that the vector were crab wasps. You would say, why crab wasps? There is a long story. During the history of wine production and science, it's known that if you take a wasp and you put it in the crushed grapes, the crushed grapes start fermenting faster. But then why wasps and not other insects? Like, say, why not Drosophila? Why not mosquitoes? Why not, ácaros? Why not, any other insect? Well, the reason was that if you look at crab wasp and you see its buccal apparatus, these big wasps are the only ones that can break the skin of the berries and therefore transfer microorganisms into the berry. So this is a study that we did a few years ago. We discovered that the abundance of Cerevisiae corresponds to the life cycle of crab wasps. We discovered that Cerevisiae is carried basically within it. This is the DNA level together with other fungi in the gut, in the intestine of crab wasps. It's also very abundant in the intestine. Is present before hibernation. Then what we did a GFP labeling of yeast. We fed them to wasps. We see if could reconstruct the cycle, and we discovered that what the wasps, the queens were passing the yeast, the fungus, to the newborn, and the newborn was bringing these microbes all over the world. And then we'd ask ourselves, then we discovered also that within the intestine of crab wasps, during the winter, when the wasp hibernate, you can find crosses between Cerevisiae and Paradoxus, creating hybrids exactly similar to those that people has found in beer. So we started hypothesizing that in reality, these hybrids were not formed during the beer making process, but were formed into the insects of the gut. To make a long story short, we are convinced, and we have evidence, that yeasts have been domesticated initially by insects and then by humans. And the reason for that is that yeasts produce volatile molecules. If you look at the volatile molecules, you see that grapes have 466 volatile molecules, wines has 644. Difference is 178. These are product of yeast metabolism. Two of those, ethyl acetate and phenylethyl alcohol, attract wasps, attract insects in general. These are byproducts of ethanol metabolism, of alcohol metabolism, and basically yeast hitchhikes on insects, uses insects to move around. And what we are discovering most recently is that when we were wild, when we used to drink fermented beverages and foods that were not sterile, we were also carriers of Saccharomyces cerevisiae. In fact, in the most recent study surveying individuals that eat fermented foods that are not filtered, produced in traditional way, we find Saccharomyces cerevisiae in their gut. Therefore, to conclude this story, we have a taste for alcohol. In reality, because probably we have a taste for several secondary products of ethanol fermentation, that yeast has evolved to attract insects and they're equally attractive for us. Yeast is a passenger, and it's a passenger that if stays as a passenger in a cycle that sees food and then us, it's healthy. If it starts invading, can also give infections, and I'm talking about other yeast, not Cerevisiae, like Candida. But we have used the yeasts in thousands of years, in at least 10,000 of years of human evolution, for exactly the same reason why insects use yeast, as a way to drink something that gives you energy. Ethanol is very energetic, also in terms of calories, as a defense against pathogen, because these yeast can produce toxic molecules and antimicrobial molecules. Ethanol is one of them, and it's a nutrient. And so maybe we did not domesticate yeast. Maybe it's yeast that domesticated us, and through the evolution of the rules of attraction that are highly conserved from insects to us. And as I like to say, this piece of history, this piece of human history, can be only understood really if we use evolution as a lens. I really believe that what Dobzhansky said, nothing in biology makes sense unless observed until light evolution, is the key factor to understand life and science. This is the end of my lecture, and I thank you for having listened, and I hope that you have understood, at least have been clear, and I'm here to discuss the lecture with you. Thank you.
There any questions? No, there aren't any questions from the audience. So can I ask a question about the role of bees, honeybees, pollen, and yeast? Do they do the same job as wasps?
This is a very interesting question. So no, they don't, and they don't for one reason: is that it's wasps can withstand yeast during the winter, during the overwintering, because they're usually carnivorous. So the sugar content of their intestine is quite low, and the problem with the honeybees is that honeybees are fed on sugars, and therefore they would get too much alcohol. It's like they would be drunk up to the level where the yeast develops toxicity. Is the reason why you shouldn't drink too much, because something that is potentially healthy in certain conditions, at low levels, as I'd say, at least because it preserves food, becomes very toxic. So this is the story, exactly the same story for honeybees. Honeybees have problems in bringing Saccharomyces cerevisiae. They have other yeasts that produce much less ethanol, and they use it as a source of nitrogen, and also to keep the beehive healthy.
Thank you. Does Ville have a question? No, Ville doesn't have a question. So I'll ask another one about the difference in the gut bacteria of African children from Burkina Faso and the gut bacteria in Western children. Anything can be done to improve our children's gut bacteria and our own?
The point is that the key strategy is acting on diet. Acting on diet. And what I'm saying, it's really these Burkina kids had a diet that was very rich in fiber and very low in simple sugars. So it's again going back to the history of cerevisia. They didn't drink simple sugars. Actually, the incredible thing is that in Africa, kids often drink banana beer, that it's a traditional fermented beverage that has like 7-8% ethanol produced by Saccharomyces cerevisiae. And even when they are 5 or 6 years old, so it's something that we would never do, but they do that. The way in which we can improve our content, really, it's mainly trying to reduce the amount of free sugars that are really harmful for the gut microbiome, and reducing the amount of meat that we eat. Because what we have seen that another reason for the selection of the Prevotellacea and Xylanibacter, so these bacteria that produce a lot of short chain fatty acids that are good for the gut health, Another strategy for that is exactly the fact that they are there because there is no simple sugars, because they divide slower. So if there is simple sugars, Escherichia coli and other gram-negative bacteria that basically like sugar a lot, they are very efficient to use it, overgrow them, and so you lose them. So we cannot think that we cannot give our microbiomes for granted. Every time we mess up or we make an error, we alter this equilibrium. And the strategy is to recover them. Then, indeed, it's to try to recover a traditional food that contains possibly microbes. And I think one of the problems as Europeans that we have been facing is the fact that we have banned some of the traditional food productions that were containing living microorganisms. And for food safety, we basically brought in place certain laws that make almost sterile foods. Sterile food are very dangerous. They're very bad for you because in a way you're missing the contribution of the right microorganism. People is understanding this, and in fact, probiotics, prebiotics. I mean,
Yes.
kefir is an example. A lot of people is starting to use them, but you have to know that a recent study showed that looking at Finland, actually, in the Baltic states, there is a divide in the rate of Crohn's disease and diabetes, type 2 diabetes, and autoimmune disorders, for which those living on the Russian side of the border are basically have a 10 times less risk of these inflammatory disorders, exactly because in the Russian part there is much more of these fermented food production that have much less regulations on how you have to keep them, and so they're very much nearer, to maybe a more traditional food rich of microorganism, and this is a tradition that we are losing because of the globalization of the food.
Thank
So
you very much. And let me check if we still have time. We only have 2 minutes, so I think it's time to say goodbye and thank you to Professor Cavalieri. And we hope to have you here in person. Maybe the Italian Cultural Institute is participating in the Settimana della Cucina, Italian Cuisine Week, and we always try to give it a scientific twist, so we can talk about wine again and maybe try a couple of glasses of wine and beer.
absolutely, that would be fantastic. I would love to come and visit you. So
Okay,
thank you for
so
inviting me, and thank you to everyone that was listening to the lecture. Have a good January.
thank you, and same to you.
Bye bye.
Bye.